Hey there, BioE 131. This is Russ giving a quick technical briefing, Module 1. This is called the Technical Briefing Module 1, Tech Brief Module 1, on how CRISPR works. I think most of you know this, so it's just going to be a review. So the key thing about CRISPR is it can be used to modify DNA. And the key elements of CRISPR are the Cas9 protein, which is this brown blob in the picture, and the guide RNA, which is the green nucleic acid shown in this picture. And then, of course, you have the genome or the DNA that you're editing. But usually we're going to be, for this ethics class, when we talk about the genomic DNA in blue that's being edited. I don't know what this blinking is, but I think it's fine. So the idea is that you have a sequence in the genome, like this sequence here that I'm underlining in red, that you'd like to modify. You design a green CRISPR RNA, which looks like this. This is a picture kind of what that RNA looks like, the green RNA, where this part is pre-programmed to interact with Cas. The little bump interacts with Cas9. This, however, is a sequence that is complementary in a Watson-Crick sense, complementary to the genomic DNA. And you can see that here because in this picture over here, you can see that the green bases, which are like these, are base pairing with the blue part of the genome. Now there is a key recognition sequence. You can see it a little bit right over here, which it says NHEJ, and that's a requirement. So you can't actually edit any arbitrary part of the genome. You have to have a part that has a G and a G with anything before it, with some constraints on what that N is. Okay, so that's the setup. We have the Cas9. The Cas9 is an enzyme, I should say this. The Cas9 is an enzyme that cuts DNA. So the Cas9 gets connected to the guide RNA. It's a guide because it's guiding you to the right part of the genome. Then the Cas9 makes a cut in the DNA. You can see the little scissor over there. Makes a cut in the DNA that is in a very specific place based on the sequence of the guide, which is complementary to the genome. Okay, so I'm going to erase all this now, if I can figure out how to do it. I think I can just circle it all. This might not work. No. Oh, erase all. There you go. So there's this picture is a little bit easier to see with all my drawing on it. Guide RNA comes into a very specific part of the blue DNA. It is complementary in that region, and there's a cut near the GG. And now you have a very site-specific cut in the genome, which is right where these little scissors are. Now there's two things you can do. NHEJ is non-homologous end joining. I'm going to let you Google that, but the point is it makes an incorrect repair that has a high chance of inactivating the gene that is encoded by that DNA. So that's a way. So one way is if you do NHEJ, that kills the function of that portion of the genome. The other thing you can do is HDR, which is homology-directed repair. And homology-directed repair, you provide a new small piece of donor DNA, you can see the word donor is there, that is sufficiently similar to the genomic DNA, such that, again, you can Google this or read it in a biological textbook, such that there is a very specific repair of the DNA in a way where you can insert what you want. Why would you do that? Well, if this gene in the genome has a mutation that makes the gene inactive, you can put in a homology-directed repair to get the DNA into a new form that is functional. And so you can fix the gene. So this one on the left side, let's change colors. On the left side, you are inactivating the gene. On the right side, you're repairing the gene. So what's so special about CRISPR? The CRISPR system. The first thing is it's extremely specific because you're providing that guide RNA, which should have only, ideally, one place in the genome where it matches. Okay, so that means you can get it to exactly the spot you want to either knock out the gene or repair the gene. It's very easy to do in the lab, easy, and it works, so that people have picked this up and it just seems to work on the first try. And that makes it a very exciting technology for genome engineering. Genome engineering. So I just want to say a couple more things, then we'll be done with this briefing. The next thing to say is that there are a couple of ways you can use this. You can use it in editing the germline, and that's the one that's ethically the most problematic, by changing the DNA of a fertilized egg. And this would be before pre-implantation. So you're about to implant a fertilized egg into a uterus, but before that, you inject these reagents, this guide RNA and the Cas9, and you modify the DNA. Someday we will probably do this, but there's a long way to go because it hasn't been fully tested. And this is the one that would be passed on from generation to generation. The other thing, which still has ethical issues, but is a little bit more straightforward, is somatic editing. In somatic editing, you're changing the DNA of an organ that is not a reproductive organ, like the liver. So let's say I have an enzyme in my liver that is messed up, and if I can get CRISPR-Cas9 into my liver cells, I may be able to repair that gene in the liver cells. However, my offspring will not inherit that change because it's not in my sperm or my egg or my implanted or my pre-implant embryo. So you can also imagine, but here you would have to get it into all those liver cells. And so what they might use is they might modify, for example, a hepatitis virus. The hepatitis virus is very good at infecting liver cells. And if you modify the virus to contain CRISPR and Cas9, then you might be able to get lots of liver cells fixed, and then you could restore function of the liver. Okay, what are the problems? Technically, the big problem is off-target editing. So I said that in general, this is extremely specific, but there can be times where the guide RNA takes the Cas9 to another part of the genome that is not intended to be edited and edits it. So it's unintended editing. And in some cases, you might edit something that nobody cares about, but you could edit biologically critical parts of the genome, including parts of the genome that when edited can cause cancer. That's the big worry, is that you're going to hit it in an area that is important for cancer, and that your off-target editing by CRISPR could lead to changes in the genome that lead to cancer. That we think we'll be able to eventually manage that by making sure that it's ultra-specific and there are no off-target edits. But right now, I think most labs are seeing a little bit to a moderate amount of off-target editing, and that's very worrisome for human applications. And that's one of the reasons that right now, this is an ethically very difficult situation. So I think that's enough on the CRISPR-Cas9 system. Thanks very much. I'm going to look up for a moment, and now I'm going to stop the recording.